Translation. So that was transcription. Remember, transcribing is copying verbatim, same language, right? Word for word. Translation is when you're changing from one language to another, like translating a message from English to Spanish or Spanish to English. Here we're translating a nucleic acid language to an amino acid language. Nucleic acid RNA message to an amino acid protein message. Okay, so we're translating to a different molecule. Now, there's this is this sort of Dr. Seussy looking thing. This is a ribosome. This green Dr. Seussy looking thing, this green hamburgery thing is a ribosome. There's two subunits, the large and the small subunit. The ribosome reads the RNA, and as it's reading it, it builds a protein chain. Proteins are made out of strings of amino acids. Right? So here's one amino acid, another amino acid, another amino acid. There's all these, there's 20 amino acids, and the ribosome knows what order to put these amino acids together based on this RNA code. Okay. These weird little bluish teal molecules are called transfer RNAs, tRNA. Transfer RNA. The job of transfer RNA is to bring the amino acids to the ribosome in the correct order. Okay, so you have, see these ones, each one, the tRNA stands for transfer RNA. So this, these are carrying, each one is carrying one amino acid, and they bring the amino acids into they're only allowed into the ribosome in the correct order. So only the transfer RNA that has the correct amino acid is going to be allowed in to pass off its amino acid. It drops off its amino acid to the growing chain, and then it leaves, and the next one can come in. And that's how they start growing. So there's 3 different kinds of RNA here, actually. These are transfer RNAs. It's a single-stranded RNA molecule that's looped up in a fancy shape on itself, and it carries an amino acid. There's this tape. This is the messenger RNA. The messenger RNA contains the DNA message that's being read here. The green ribosome is made out of a conglomeration of ribosomal RNA, rRNA, and protein mixed together. So the ribosome is actually an enzyme that's not just made out of protein, it's made out of a conglomeration of protein and RNA. Okay. So different RNAs have different functions. All RNA, every kind of RNA out there, its job is to express the genetic code as protein. But there are different RNAs have different, they take on different roles in the expression of the genetic code. So you have the genetic code in the DNA. Messenger RNA is the photocopy that carries that message to the ribosome. The ribosome is an enzyme made out of RNA that reads the genetic code and builds a protein from it. And the transfer RNAs are an RNA molecule that carry amino acids in the correct order to the ribosome. All right, so just again, quick recap. What are proteins like enzymes made out of? What are they made out of? So this is from prereqs. This is something you really have to remember to understand transcription translation. Proteins and enzymes are made out of amino acids. Proteins are made out of strings of amino acids. What do proteins do? So I don't have this on a slide, but I will list them. Proteins can be enzymes, which we've covered extensively. So proteins are strings of amino acids folded up in a 3-dimensional shape, and they have functions. They can be enzymes. Enzymes are proteins that catalyze chemical reactions, that facilitate chemical reactions. They can build large molecules out of small ones. They can break large molecules down into smaller ones. Okay? So enzymes that conduct chemical reactions, that's a kind of protein. Other proteins can be transport molecules and portals that allow substances into or out of the cell, across the cell membrane. Those are made out of protein. Proteins can be structural. For example, the cytoskeleton is made out of protein that holds the cell's shape. Proteins can be communication molecules like hormones or neurotransmitters, or communication molecules between bacteria. Okay. Proteins can be defense molecules like antibodies and other antimicrobial peptides. Okay. So those are, let's see, is that the main thing? So structural, portals into and out of the cell, enzymes for chemical reactions, defense, communication. I could also say movement, because usually the flagella or cilia or even the cytoskeleton, you know, those are all involved. They have these proteins, marionette strings that help them move. Right? So there's just lots of different protein functions. So DNA codes for proteins, and proteins do everything for the cell. They can build plasma membranes. Proteins, enzymes build plasma membranes. Proteins are in the form of enzymes build vitamins, right? So the DNA codes for protein. Protein does everything else in the cell, right? So, and proteins are strings of amino acids. So you have nucleic acids coding, strings of nucleic acids coding for strings of amino acids. Okay. Steps of translation. And we'll cover this, and I know we had a picture. Now I'm going to cover this in text, then I'm going to have another picture, another couple pictures. First step, messenger RNA and transfer RNA carrying its amino acid are brought together in the ribosome. So you have this ribosome, the messenger RNA with the gene in it is put into the ribosome. Transfer RNA carrying an amino acid is also put into the ribosome. So the three kinds of RNA plus the amino acid carried by the transfer RNA are all brought together inside the ribosome. The messenger RNA and transfer RNA are matched together in groups of 3 nucleotides. So you can think of the genetic code as a series of 3-letter words. The 3-letter word is called a codon. So let's say it's AUU, right? Because, sorry, RNA has U, not T, right? So AUU. That's a codon. That's a 3-letter word. That has to be matched to a complementary anticodon in the transfer RNA. So if the codon in the messenger RNA that's in the ribosome is AUU, the complementary anticodon in the transfer RNA is going to be UAA, because A's and U's match up with each other. So the transfer RNA with the codon is AUU. The anticodon is UAA, so the transfer RNA, the transfer molecule with UAA is brought into the ribosome because it matches, and its amino acid is put onto the building protein chain, the building amino acid chain. Once the transfer RNA drops off its amino acid, the transfer RNA is released, and the next transfer RNA molecule can arrive and add its amino acid to the growing chain of amino acids. So note, every transfer RNA carries a 3-nucleotide code called an anticodon that codes for a single amino acid. So let's take a look at how that looks. And I also have videos of this if you need even more review. So here, this red thing is the ribosome, large and small subunits. Here's the messenger RNA with the photocopy of the gene, the blueprint for this protein that's being built here. This is all about building a protein that the cell needs. So the instructions for the protein were in the DNA. This is the messenger RNA is the RNA photocopy of that gene. It's brought to the ribosome to read it. The ribosome's reading the photocopy of the blueprint for this protein and building from it. So here is a codon. Here's another codon. Codons are 3-letter words made out of nucleic acid, made out of nitrogenous bases. So this codon is ACC. The only transfer molecule that is allowed to fit into the ribosome at this point is the transfer molecule, the transfer RNA that has the complement, the complementary anticodon. Here's the codon, here's the anticodon. They have to be complements to each other. A's to U's, C's to G's, C's to G's, right? This is the only transfer RNA that can be there. The transfer RNA with the UGG anticodon always has the same amino acid. Let's just call it guanine. I don't know which amino acid it is off the top of my head. Let's just say it's guanine. Okay, so guanine gets added to the growing polypeptide, the growing protein chain. Second transfer molecule brings its next amino acid in here. This is the only transfer molecule that's allowed in because it's the only transfer molecule with a complementary code. This codon is AAA. The complement is UUU because A's and U's are complementary. So transfer molecule with anticodon UUU comes in. It's bringing an amino acid. Let's just say it's leucine. So leucine then gets added. So in this way, the codon determines what anticodon can be there. The anticodon determines what amino acid is brought. And so that determines the order of amino acids in this growing chain. So let's take a look at this transfer RNA molecule. So I told you that all RNA is single-stranded, but I told you that it can loop up on itself in these fancy shapes. So these sort of have like a little bit of a crucifix shape, right? And this shape is held together. Here's the anticodon. This is what matches with the messenger RNA in the ribosome, right? But this is the way this is drawn is a little bit deceiving because it makes you think that this is the only place where there are nitrogenous bases. This is a string of nucleic acids. Nitrogenous bases are all over the place. In fact, it's nitrogenous bases that hold the structure together. So it says complementary base pairing here. Maybe you have A here, U here, A's and U's bind. That holds these folds together. So it's complementary base pairing internally that keeps the three-dimensional shape. But then these are flipped out for the anticodon to match with the messenger RNA in the ribosome. So this part just keeps its shape. This is the only part that matters for translation, for translating the nucleic acid code to a protein code, an amino acid code. And here's the amino acid that it carries. So let's look at that again. So here are this transfer RNA molecules, and here's the messenger RNA carrying the gene. So you can see how the anticodon matches up with complementary base pairing to the codon, drops off its amino acid, then it's going to leave. So I have a couple videos that you can watch on to remind you. Actually, I'll just do one that has, well, one of them is going to have everything from DNA replication, DNA supercoiling, and also transcription translation. Then I'll have a shorter one that's just transcription translation. So if DNA doesn't make any sense to you, you don't remember any of this, I suggest you watch the longer one. This one really is a more basic one on transcription translation. Also, the video quality is better on this one. It's only 4 minutes long. All right, so let's look at this letter by letter. So we already looked at, here's the template DNA strand. The template strand is the one that's being read by RNA polymerase. RNA polymerase unzips these two strands to separate them. Then it reads the template strand. It knows which one is the template because of the promoter sequence that precedes the gene. So it reads just the template strand. It reads it in the 3' to 5' direction. It builds a complementary RNA strand through complementary base pairing. Now, you can break this down into words of 3, which are called codons. And we have cracked the code. So you can look at this and you can figure out what protein is made because we have these codon charts. So you'll have an exercise before the exam where you'll get to practice this. So what you do is, okay, let's start with this first codon. Oh, and just to make things even more confusing. So I told you that DNA, when enzymes that read DNA always read 3' to 5'. The ribosome reads RNA 5' to 3'. So 3' is the front in DNA, 5' is the front in RNA. So here is AUG, 5' AUG. So we're going to take first position in the codon is A. So let's look at this codon chart. Okay, on this side you have GACU. This is for the first base, the first position in the codon. Well, we said it was AUG, right? So here's A. So we're going to look at A. This whole row is all the codons that start with A. The second base, second position in the codon was U because it was AUG. So here's the letters for the second base, U. So we want the intersection of this column for U and this row for A. So that's these. And we can look at, so now we've narrowed it down to 4 and you can see AUG right here. AUG stands for methionine and it's also the start codon. Okay, this is how the ribosome knows where to start. So this is the start codon, it's also methionine. So AUG, the codon in the RNA, AUG always codes for methionine. Let's do a second one. Second codon, CUA. CUA. So you find C for the first one, there's C. Here's U for the second one, so here's all the CUs. CUA is for leucine. Well, you can see that CU anything. CUC is leucine, CUU, so CUA is leucine. So we have methionine, then leucine, GGU. First one is G. All right. First base, G, so we're in this row. Second base is G. Here's the column G for the second base. GG, what did I say? GGU. Okay, so GGU, glycine, right? And then finally, so we have methionine. what did I say? Methionine, leucine, glycine, and now CAG, the last one, CAG. So here's CAG. Okay, CAG. Honestly, I don't remember which one this is. And I'm not going to ask you to remember any of this. I want you to know how to use a codon chart. I'm not going to ask you to memorize any of this. Okay. So now we have the code. Polypeptide chain, methane, leucine, glycine, it doesn't matter, like whatever GLN is, you can look these up. So, you know, I haven't even memorized all of them, right? It doesn't really matter. All that matters is that you know how to use the codon chart, because once you have these amino acid chains, that determines the structure of the protein. Because remember, we talked about how the when we talked about metabolism, we had a section on enzymes, and I told you the three-dimensional shape of the enzyme is essential to its function. That's true for all proteins. This order of amino acids determines the three-dimensional shape, because each of these has a different chemical property. Some of them are positively charged. Some of them are negatively charged. Some of them are attracted to each other. Some of them repel each other. Some of them are attracted to water. Some of them are hydrophobic and cluster inwards away from water. So those chemical interactions between these amino acids in this one chain, this chain is going to be longer, right? They're going to be hundreds to thousands of amino acids long. They're going to fold up on each other in these complex three-dimensional shapes based on the chemical interactions between each individual amino acid. Okay. So there is an activity. I'm going to put it in the exam folder, and that will just help you to understand transcription translation. I think the only way to truly understand it is to do it yourself. Okay, so I really recommend doing that. So this whole process, this is gene expression. Here's the gene in the DNA. Here's the expression as protein. This is the physical outward manifestation. This is going to be a protein that produces melanin for brown eye color or brown skin color. This is going to be a protein, and it's either going to be functional or non-functional. If it's non-functional, you don't get brown color. If it's functional, you do get brown color, right? It's going to be a protein that, like hemoglobin, that grabs oxygen and transports it through your body. You know, this is going to determine your body chemistry, how you make neurotransmitters, how you make hormones, what color your skin is, what texture your hair is. These proteins determine all of that. So they determine your physical characteristics, genetic code, physical manifestation. Okay, so that's what we call gene expression. The transcription and translation put together is the expression of the blueprint, the outward expression of the blueprint. It's how it happens. Okay.